Hear me okay now? Okay, awesome. You can hear me better if you take your headphones off. This is my daughter Peta and my mom. They're a great support network here tonight, and thank you all for coming. Just a little bit about me in the event that you just showed up and you have no idea who I am or what I'm on about. My name's Jody Gerdts. I'm from the States, but I've been here, since 2013, so I know a lot of you in the room, so thanks for showing up tonight. I've been beekeeping nearly 20 years, involved with research, bee breeding primarily, and I've been on this kind of like track of how do we prepare for living with Varroa in Australia ever since November 29th, 2013, when I stepped foot into Australia. Even before that. So I've been really fortunate. I've done my PhD here studying chalk brood, worked a lot in bee breeding space with hygienic behavior, and I've been awarded 2 study fellowships. And a lot of that information I'll share with you tonight. And the question on both of my study fellowships is, how do we breed for Varroa resistance? All right, and now we've kind of maybe, hopefully, have this incursion contained, and it's not such a hot topic as it was perhaps 6 weeks ago, but there's a lot of information and a lot of misinformation out there that I just wanted to plant some seeds, sow some seeds, and get you guys kind of thinking about what's happening in the world, what might be possible here in Australia, and what you might be able to do, you know, even within your own beekeeping operations. So I'm just going to go back one quick. I called it a bee's eye view, living with Varroa, a bee's eye view, because we are so egocentric and we just keep thinking about it's us that's living with Varroa. It's not actually us. We're beekeepers and we keep bees that are living with Varroa. So just if you kind of change your mindset a little bit about whose problem is it, then we can start thinking about how the solutions come about. The way the program will go tonight, the 2 fellowships that I recently received, honeybee, or the Varroa and honeybee life cycles, flattening the curve. You thought maybe we were done with that topic, but here we are, flattening the curve, or trying to. Selection for traits, and then a summary and concluding thoughts. And that's when I get to stand on my little soapbox and tell you what I really think about things. So 2019, I was awarded a fellowship from the International Specialized Skills Institute. And the title, Investigation of Varroa Resistance Honeybee Breeding Programs in the United States for Application in Australia. And I went to a whole bunch of different places, but the main areas of interest were Louisiana, so the USDA lab in Baton Rouge. That's a honeybee breeding genetics and physiology lab. Up here in Indiana at Purdue University, and then kind of working in with the Kentucky Heartland Breeders Cooperative. So at the USDA here in Louisiana is, have you guys heard of Russian bees? Yeah, Russian bees. Okay, so they're touted as being resistant to Varroa because they've been living with Varroa longer than the rest of Apis mellifera. So the USDA lab brought in a bunch of Russian bees and said, can we have bees that are resistant to Varroa? And then yeah, maybe kind of. And then they started breeding VSH. And then they started another breeding program. And I'll tell you about what VSH and that kind of breeding program is in just a little bit. But basically, they've been involved with breeding for Varroa resistance for the last 20 years. Okay, up in Purdue University, they're working on mite biter bees. Have you guys heard of this? Mite biter bees, ankle biters, grooming behavior. Cool, because I'll tell you about that today, or tonight. So in these 2 groups, work really closely with one another. I'm just setting you up for more information later. And so where are we? We are in October. So it was just in July and August. I went over to Europe because I thought, okay, I learned all of this stuff in the United States, but I just needed to know more. I've been working with the Arista Bee Foundation for the last 4 years, and they're based in the Netherlands, and learning all about some cool breeding work that they're doing with their cooperatives in Europe. And I thought, well, I need to go and see boots on the ground, what's happening there. So investigating commercially viable, locally adapted honeybee breeding practices in Europe with a focus on pollination and Varroa resistance for application in Australia. So you can see how the focus is evolving. It's not just about Varroa resistance. Yay, that's where we're going. No. We're going commercially viable, pollination-specific bees. Because there's a lot of bees, and you'll hear about this coming up. There's a lot of bees that can handle mites. But you guys probably don't want to work with a lot of those bees. So that's the trick. That's what we're up against. So in Europe, I was up here. These are the green arrows are the programs or the groups that I met with within the Arista group, okay? And then down here, this is ENRA, the National French Institute of Agriculture and Technology, which I spent a week there learning about the things that they're doing. So I'm just kind of setting the stage. Man, I've been everywhere asking a lot of people this question. How do we breed for Varroa resistance? So now we're on, in Varroa resistance, okay, cool, let's talk about the Varroa mite, what's the life cycle, and how it relates to honeybee life cycle. It's pretty complicated, but basically, how many of you, I just want to see a show of hands, how many of you feel like you really understand what the Varroa mite is and how it reproduces on our bees? Okay, cool, great, this is great. Okay, so basically the really challenging thing about Varroa mites is a female foundress, so one mated female mite can start a whole new population, a whole brand new population. It just takes one mite because brothers mate with sisters. Okay? So what happens is just as, this is called fifth instar in the developmental stage of the honeybee, so just as the cell is getting capped, that mite respond. It doesn't have any eyes, okay? It responds to chemical cues in a dark beehive, and it knows when this cell is just about to get capped, and it sits down in the jelly, the worker jelly down here, sends this little periscope, little tube, breathing tube out, and just waits until the cell gets capped. Within about 5 hours, a feeding site is initiated on this prepupa. So she starts immediately feeding on the prepupa, and then starts at 70 hours, she lays a male egg, and then 30-hour intervals after that, she lays female eggs. So that male egg matures first. So as his sisters mature and they're receptive, they start mating. Okay, and it happens all within the confines of this worker cell. And it can happen in a drone cell too. The really great thing about a drone cell is it takes just 3 days longer to emerge. So on average, it's about 2.1 reproductive mites come out of a drone cell, where as a worker cell, it's 1.6 reproductive mites in an uncontrolled population. So you see all this reproduction and all this mating happening, and basically that picture is a bit, I wish there weren't so many mites coming out, but basically there's the foundress that went in, comes out, and then 1 or 2 reproductive mites, other foundresses, come out, okay? And so what happens is we get an exponential growth curve, right? So in relationship, if you guys are interested in cool beekeeping stuff, check out Randy Oliver, scientificbeekeeping.com. His mind is amazing and he goes and goes. But basically, as the bee population increases, you can think spring buildup, summer height, the mite population also increases. And as the bee population in the autumn decreases, the mite population is at its peak. And so that's where you have basically every cell or every one of your winter bees has been parasitized along the way. And the hard thing about Varroa, it feeds on not on the hemolymph of the bees like we thought for a long time, but actually feeds on the fat bodies of the bees. And the problem with feeding on the fat bodies is that that's where the bee's immune system exists. That's where the immune system functions. It's in the fat bodies. So with the reduced fat bodies, and in all the other parts of the world it vectors viruses as well. We don't have that viral landscape here. But it's just bad news for bees, okay? But we start thinking about resistance, Varroa resistance. What does that mean? Well, the act of fighting against something that is attacking you. And that's basically, it's not about what we're doing, is it? It's about what the bees are doing. And they've come up with great strategies to defend themselves against these mites. You can see up on the top, the red ones are the female reproductive mites. The next row is when they're just starting to harden, starting to mature and sclerotize. The very, very top, I think, are that's the male. It's all in German. That was the hard part about traveling in Europe, is I only speak English. So there's a lot of things that I have that are in different languages. All right, here. And now we come to the part about flattening the curve. Why is it important? Let's see if we can get back here. So with COVID, we thought about flattening the curve because then we can keep the infection rate below that which our healthcare system can manage. Same thing with mites and honeybees. Keep the population below that which the bees can manage. The bees have lots of different ways to manage their health. That's not actually what this presentation is about, but I can tell you about that some other time. So we're flattening the curve, and the different strategies that bees have come up with to flatten the curve. So there's this VSH, Varroa sensitive hygiene, SMR. The bees didn't make up these names, we made them up. I don't think if you asked a bee, how's your VSH going? I don't think they, what? SMR, suppression of mite reproduction, grooming and biting, and broodless periods. Okay, so in Varroa sensitive hygiene, it's a really highly developed type of hygiene in response to chemical cues. And again, this is in a dark environment of the bee nest, but workers detect, uncap, and remove if it's really bad infections or otherwise interfere with mite reproduction. Before the mites harden and sclerotize, they're really wimpy, right? And just even a little bit of exposure to air will dehydrate them and render them, hi Chris, render them useless. So then we have SMR. So this VSH, You might hear a lot of talk about VSH this, VSH that. But VSH, just think about it this way. This is what the workers do. This is an adult bee behavior. But then we have SMR, which is a suppression of mite reproduction. And this is where mites, for some reason, they fail to reproduce on the pupating bee. Talk about this in a little bit. Grooming and biting, where bees actually groom the mites off and bite their legs and just give them a really hard time. And then broodless periods. Why do colonies go broodless? When? In what situations do colonies go broodless? Meaning don't have brood. Queenless, supersedure. What else happens? Winter conditions, so response to environment. Swarming. Been sprayed. Yeah, but that's not something that the bees control. Just think about it this way. How good, from your perspective as a beekeeper, are broodless periods? Yeah, they're awful. They're terrible. So when we think about breeding for Varroa, this is how to get the bees to do what we want them to do and manage Varroa. Broodless period doesn't really factor in too much in our breeding perspective. But it does, and it needs to factor in our management perspective. So flattening that curve, the bees have behavioral and biochemical means. Beekeepers have chemical and mechanical and genetic ways. We're not talking about what beekeepers are doing. We're talking about what bees are doing tonight. So selection for traits. This is the breeding side of it. So selection for Varroa-sensitive hygiene. And remember, this is where we as beekeepers step into the scene and say, okay, which of you are doing good? Which of you are managing Varroa? We want more of you, and we're going to replicate you. But the most important part about breeding for Varroa-sensitive hygiene is you have to start with good stock. You have to start with productive, commercially viable, honey-producing, gentle stock to start with, and then you can work on it and get some Varroa resistance on the other end. If you start with highly resistant bees, good luck getting them to make honey. It's hard to add those good traits back in, but you can add one trait into something that you already like. And this is what the world is finding. So going back to the USDA lab in Baton Rouge, and I'll just tell you a little bit of a side note story about this. They imported Russian bees. Oh yeah, Russians are really good because they're really great at handling Varroa. But the thing is the moment any sort of a nectar flow shuts off, well, so do they. They stop laying. They go broodless. They also swarm quite a bit. Well, that's not really good. That's not. The commercial uptake was horrendous. So then they started just selecting, doing mite counts and selecting from the lowest mite colonies. And they ended up with this line of bees called VSH. Great. Well, they were just selecting for low mite counts. They weren't selecting for honey production, or they were selecting for temperament because they had a lot of, novice people working on these bees. But at the end of the day, commercial uptake was crap because they weren't commercially viable bees. So then they started, okay, we need the pole line bee. So they started with commercially viable stock, overlaid all the selection methods, which are some of this stuff, onto commercially viable stock. And now, within the last 4 or 5 years, have that stock under massive field trials, going to almonds, going to North Dakota, going to Florida, all over the United States. And they're actually doing pretty good. So this Varroa-sensitive hygiene is where those worker bees, those adult bees, can detect a reproductive mite within a cell and open it up. And the way that you select for VSH is you have to open up all those cells and count how many mites, if any, are in the colonies. And if the colony is really heavily infested with mites, well, that's job's easy, not resistant. But otherwise, it might take you 2 hours to get through one of these frames. Keep that in mind when you think about, oh, I'm just a hobby beekeeper. There's nothing really that I can do. There's everything that you can do, because that's who's doing it. It's the people that have the time and the patience and the skills to do this type of stuff.
Jody, if I may,
Yeah?
wouldn't an alcohol wash give you a similar result?
No, because we're looking for infestation in the brood cell.
But if you did an alcohol wash and you only got one Varroa mite, it's indication you've got a fairly good queen or a fairly good resistance.
Yes, no, there's probably a lot of ways to shortcut this. I'm just saying what people are doing in the world. So hold on, we'll talk about it at the end, okay? Thanks, Chris. No, that's okay. So the Arista Bee Foundation has set up this network of people just like yourselves all around Europe that are looking for colonies and assessing single drone inseminated colonies for this type of behavior. Because it's about being able to isolate and get really closed in on that specific trait and then outcross it to keep your productivity. It's massive, it's big. I'll show you some more pictures about what people are doing. In France, they're looking at chemical cues of Varroa-sensitive hygiene. So what is the chemical that bees are responding to? Can we synthesize that chemical? Can we test it on colonies? And can we potentially select and breed for resistance without having to do that really tedious, laborious method? This is an example of the type of work that has been happening in Europe selecting for VSH. These are all single drone inseminated colonies, and this is one of probably 15 groups that had probably 40 or 50 people just like yourselves at assessment day looking and counting mites, looking for and counting mites. So it's very, 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 very inspiring. Moving on to the suppression of mite reproduction, which works hand in hand with this VSH. The VSH is the adult behavior. The suppression of mite reproduction is pretty poorly understood, but it is something that the pupating bee does. So not much is known. There's biochemical signaling, but here we see VSH bees. We're looking at non-reproductiveness in the green. VSH bees, when looking for VSH, are also seeing the mites not reproduce. And this is just a picture of a printout on a wall at NRA. But basically, they were artificially rearing mites and putting in order for the mites to reproduce, they need the chemical signal of the pupating bee, just even in the vicinity of the mite in the petri dish. And when they put the replicating or the mites that wanted to reproduce next to highly resistant bees from highly resistant colonies, they didn't reproduce. But when they put the mites in the petri dish near susceptible colonies or susceptible pupae, they reproduced. So there's some chemical signaling, there's some biochemical pathway that is just like, oh, this is really cool stuff. This is kind of like happening right now. The investigation anyway. And And this is another project that is just so cool and so inspiring along the lines of the mite grooming and biting. This is the Purdue Lab, Purdue Bee Lab, plus the Heartland Honeybee Breeders Cooperative, and this group, Sustainable Genetics Transfer Network. It's all got some pretty flashy stuff, but basically it's a whole bunch of people just like yourselves that are doing these sticky mats, looking at the mites that fall down, counting how many mites out of 100 have been chewed, and sending the semen or queens or some sort of genetic exchange across states, across beekeeping clubs to create lines of bees that are resistant due to grooming. Now, they're not actually looking at the VSH behavior as well, but they're looking at the grooming behavior. It's another way that bees are able to handle this, right? Again, it's very, very tedious, but it's going to show results. Is that okay?
It's not going through the zone.
Oh. And then it's not going forward. VSH, SMR, grooming and biting, you can select for that in any population, in any group of bees. Can we?
Try that.
Go forward. There we go. There we go. And we talked about broodless periods. This is a Russian bee. Talked about the times that we have broodless periods. I don't know about you guys, but ooh, my brood nests look pretty sparse at the moment. It's a bit honey bound due to a bit of canola involved. Swarming, plugging up brood nests, superseding, queens go off laying. All well and good. And when you start thinking about Varroa management strategies and chemical application, these are the times that are the best times to put your chemicals on, right? Because you've got broodless periods. But from a breeding and a beekeeping standpoint, not terribly helpful. So bees, it's been shown so many different populations of bees all over the world, isolated populations of honeybees have developed resistance, have been able to live in peace and harmony and balance with this mite, okay? We are the problem. Beekeepers are the problem. We get in the way. And we get in the way really in two ways. We use chemicals to artificially prop up the bees, and we have high density apiaries. So when you think about the bond test or just letting colonies crash and burn and die and become mite bombs, well, that doesn't happen if bees are living in their natural density, because the highly virulent mites kill the colonies and then they've got nowhere to go. But what happens here, we've got highly virulent mites and highly virulent viruses that kill colonies, and then they get robbed out, and then they have another home, and then they just go somewhere else. So we are actually the problem. Breeding programs around the world also have found a balance. And the problem is we've been focused so long on just the mite problem, keeping the mite populations low, that we've actually forgotten that we need to make honey. So we can keep selective breeding pressure on resistance to other diseases, honey production, temperament, the things that we're interested in, and Varroa resistance. So we can, and it's happening, like it's happening in the United States after a long series of failed attempts. They're not failed. You learn something every time you make a mistake. and in Europe, there are commercially viable Varroa-resistant populations. And we're actually on the ground floor of that movement. So I think that's very, very exciting. Just a few pictures from France. This is a whole big beekeeping group that's working with the university that's looking at Varroa populations, hive health in general. So then they're pooling the best of the best colonies to use that as their breeding stock. Pretty cool, pretty innovative. This is the autumn hive assessment. So this is just after the lavender flow, and it's a honey-bound brood nest, and this is when they put their Varroa treatments on. And that kind of got me thinking, oh yeah, we don't need winter. We've got plenty of honey-bound brood sessions amongst us. This is a group out of the Arista program, also in France. A family that's been keeping bees for a very long time and are dedicating their lines of bees to breeding for resistance. These are just pictures of, a young female mite and a young male mite. That's the kind of thing you're looking for. Sorry. This is in Germany. Again, like I said, this is 1,000 people just like yourselves that have come together to learn about the bees, learn about the mites, and try and find, the light at the end of the tunnel, and they're having a lot of success. This is in Switzerland. And that's it. So I reckon we're super fortunate in Australia because people have been at this for a very long time. Very skilled and very talented people have been at this for a very long time. And we get to use all of their gains and all of their mistakes and start off on a pretty fresh space. So just know that it's not something that happens in the bee breeder's hands. It's not something that somebody else does. It's something that all of us can do in a coordinated effort.
Thank you, Jody. Do you have any questions for Jody at all?
When you showed the two male and female Varroa on the back of the bee, we usually thought that they're on the head. Do they shift up from there to those little creatures?
Yeah. So this is, this pupa has just been taken out of the cell. So this is what the mite family looks like on the bottom of the cell. Yeah. So you're right, the phoretic mites, the red adult female mites, travel generally on the back of kind of like nurse bees age, but this is what's happening on the inside in the nursery. Yeah.
Anyone else?
A while ago I saw a YouTube clip, I think it was Professor Thomas Selby, who had his thing was the Darwinian theory.
Yeah.
Would that work in the long run if we just kept out of it, or is that likely to be just too decimating?
No, it's going to be terribly decimating. Yep. And we're not going to have a commercial honeybee population, or we're going to end up with bees that we actually don't want to work. So it will happen. It will happen on its own, and I'm speaking from the standpoint from a viable commercial industry, like just letting them die is just not good enough, especially when we've got lots of tools and selection methods and ideas to be able to get somewhere.
So he's not a popular thought?
Oh, he's brilliant. Tom Seeley is a brilliant researcher, yeah. But I think on a commercial scale, we are doing something so not natural with our bees. I mean, let's face it. So we can learn from what they're doing, but at the same time, I just can't see anybody actually doing that.
Anyone else at all? No. Chris?
Jody, listening to your talk, you very much emphasise the cooperative effort that goes on in Europe and these places. And when we're dealing with Varroa mites, we're dealing with very big numbers and a lot of work, such as uncapping a complete frame. It takes hours to do. Could Australia learn now and start to organise cooperative groups ahead of it? Is there any movement in Australia for this to happen?
Chris, yeah.
What can be done about it?
Yeah, no, there's absolutely movement for it. And we're just...
What is this movement? I'm a beekeeper, I haven't really heard of it.
So there's ideas coming about. There's Plan B, right? So this is the National Breeding Genetic Improvement Program, but there's also smaller scale individuals and businesses that are starting to come together. It's all about drone selection. It's all about funneling your best genetics back into a place where it can be replicated and distributed for evaluation. David and I are working on some ideas. We spoke at the VAA conference. Some of you guys were in this room, were there. So it's happening. It's slow. We're very busy. We're all very busy people. But I think the more we talk about it, and the more we...
It's very confidential.
Sorry?
And I get the feeling it's very confidential.
I don't think it's confidential.
Well, I reckon I read, and I'm not reading very much about it.
No, it's just, it's grassroots, Chris. It's just up and coming. It takes all of us to get together and do it. It's not something that somebody else is going to do, you know?
Thanks, Jody.
Yeah, thank you. You can have both of them.
Thank you.
Sure.
Just on
Yeah, I'll just...
the Bendigo branch, we'd like to thank Jody very much for her presentation on time tonight. We really appreciate the time you've given.
Sure.
I can see by the way people are watching and listening and observing that they've also been very enthralled and interested in what you're doing. And we'd like to accept this small gift, Bendigo gift.
Thank you.
For coming tonight. And
That's lovely.
We hope we get you, hope your little daughter's not too asleep now.
No, not yet.
So thanks very much,
Yeah,
Jody.
thanks, Kel.
That's
Appreciate
great.
it.
We'll see you
Thank...
when we see you.
Yeah, that's right. Thank you, guys.